In 1997, a French woman named Jane Calment passed away at the age of 122 years and 164 days, making her the oldest person in the history. She was born a year before Alexander Graham Bell patented his telephone and 14 years before Eiffel Tower was built. And one of her interviews she said that she was 12 to 13 years old when she saw Vincent van Gogh. So this video essay is about longevity and how to reverse aging. Reverse aging doesn't only mean to live longer, the important thing is to live healthier while aging. You can increase not just your lifespan but your health span. The prevention of aging is about adding more years of healthy life during which you are free of disease and energetic and you have an enthusiasm for living. Longevity means extending youth, not extending old age. David Sinclair, an Australian biologist and professor of genetics, will tell you this clearly.
If you look at this graph, actually, this really brings it home. Now, we're always taught that our medicines are making us healthier for longer. That's not true. Look at this graph. The amount of time that we are spending in good health is actually decreasing in terms of percentage. No wonder healthcare costs are going up. What we need to do, of course, is to keep us healthier for longer. So I'm not talking about living for 500 years. I see that sometimes quoted in the press. But what I am talking about is an ability for us to live into our 90s and our 100s in a healthy way.
What do we know about longevity? Let's start from the beginning.
So one of the big questions in biology is, why don't we live forever? Maybe one day we will, but until we understand why we grow old and get diseases such as Alzheimer's, diabetes, cancer, cardiovascular disease, we will not have a chance to greatly extend our lifespan.
Why do we age in the first place? In 2013, a group of researchers from all fields of science which are related to aging bring all together a combined theory of aging called the hallmarks of aging. There are many theories explaining the aging process, but one of the oldest, well-known and most researched theories are mitochondrial theory of aging. Mitochondrial theory of aging, it was first introduced by Denham Herman in 1950s, which he later expanded to the mitochondrial free radical theory of aging. In this theory, the aging is caused by decrease in mitochondrial function.
The idea is that as the mitochondria, when we're young, are functioning, they're producing these free radicals that damage the enzymes, and particularly the DNA within the mitochondria. And over time, we start to lose the code that's in our mitochondrial DNA. We get mutations, we get big deletions. So for example, if we took my DNA from pretty much any cell, except perhaps my germ cells in my testes, these cells would have a large number of mutations and deletions already, which is rather disturbing.
What actually happens in the cell can be described by Dr. Christian
As we
Leuvenberg.
age, or even as we're young, proteins and DNA become damaged and they turn over. So we need very healthy proteins and peptides and DNA and RNA. And due to a variety of stresses, from oxidative stress to glycated stress, reactive nitrogen species, so different forms of stresses can damage those small peptides or proteins. So we need to remove them quickly to get new healthy ones in place of those, so we can optimally function in the cell.
Due to these processes, the changes happen in body while aging are: first one is growth hormone decline. The growth hormone decline will cause of sarcopenia, which is process of declining of body mass, muscle mass and strength. As we age, we start losing our muscles mass and strength. Third one is bones and joints. Density of bones decreases, loss of cartilage and stiffening of ligaments and reduces joints mobility. Fourth one is slowing down of brain function because of decreased neural activity. Recent evidence indicating that nutritional, pharmacologic and genetic manipulations to extend lifespan and or health span often or possibly always stimulate autophagy, and that increasing autophagic flux mechanistically contributes to the extension of longevity. Which brings us to the second part of the video, which is autophagy and how it is connected with aging. What is autophagy? It is totally normal if you haven't heard of this word. Christian Dedube, a biochemist who introduced the word autophagy. He made beneficial discoveries of two-cell organelles, peroxisome and lysosome, for which he got the Nobel Prize in Physiology of Medicine in 1974. The technical autophagy definition is the maintenance of the nutrition of the whole body by metabolic consumption of some of the body tissues, and the segregation and disposal of damaged organelles within a cell. Better explained by Christian Dedube himself.
And autophagy is a self-eating by cells. It's a mechanism whereby small bits of the cell are segregated. It's a complex mechanism, segregated within a membrane and then conveyed to lysosomes for breakdown. So this is autophagy. Self-eating is one of the mechanisms whereby cells degrade their own substance and replace it, so it's part of turnover.
When lysosome was first discovered, scientists were examining a rat liver and added glucagon, a peptide hormone, to it. The researchers found that it resulted in the degradation of mitochondria and other structures within the cell. This is the first recorded viewing of glucagon-induced autophagy. This finding stimulated interest in the physiological role of autophagy. Later, Yoshinori Osumi, a Japanese cell biologist, discovered how our body degrades and recycles its cellular components. He got the Nobel Prize in 2016 for his discoveries of mechanisms of autophagy. During autophagy, a cell transports its broken down components to a certain area where they are taken apart so that their chemical components can be reused. The autophagy process is multifold, and there is a variety of proteins involved that create this garbage back in the cell. To take these larger molecules or damaged mitochondria and remove them and carry into lysosome for lysosomal degradation. They fuse with the lysosome and the organelles are recycled and they can be reused in the cell, so it's a self-eating process. To understand it in easiest form, let's go back to Yoshinori's talks, in which he was talking about protein degradation in plants. All the protein in leaves degraded completely and all the amino acids go to form the grains of the rice. So degradation is most important in making of the next generation. Degradation is not negative way, it is very important.
The reason leaves turn yellow, that means that all the protein in the leaves are degraded completely and all the amino acids go to the this grain of the rice. So for rice, degradation is very important to make next generation these grains.
Two major roles of autophagy are, first one is nutrient recycling, which is essential for survival under starvation, and the second one is elimination of excessive or harmful materials like essential for clearance of cytoplasm, selective degradation of specific proteins, protein aggregates, invasive bacteria, virus particles. And how is autophagy connected with aging? Autophagy promotes the survival of healthy cells. Everything you do in daily life, walking, talking, working, or even sleeping, these things are causing some kind of oxidative stress and they are causing damage to mitochondria. And if not controlled, it will eventually lead to apoptosis, which is death of cell and destruction of mitochondria, which is the main reason of aging and getting disease related to aging, like cardiovascular disease, cancer, arthritis, cataracts, osteoporosis, type 2 diabetes, hypertension, and Alzheimer's. On the other hand, increased autophagy helps in controlling the cause of this disease. Autophagy could promote longevity by a number of mechanisms. It actually helps to clear toxic proteins and defective mitochondria. It suppresses oncogenic transformation and could help maintain stem cells, boost immune function, and possibly regulate insulin homeostasis. You can find many researches done which are directly indicating that autophagy is connected with many diseases and can control or even cure many of these. Because autophagy can remove protein aggregates which are associated with the most neurodegenerative disease. In Alzheimer's disease and Parkinson's disease, brain's autophagic and lysosomal vesicles accumulate, suggesting a blockage in the autophagy process. The toxic proteins associated with neurodegeneration might directly inhibit autophagy. Mutants in autophagy itself promotes protein aggregation and neurodegeneration in various model organisms. Autophagy also removes defective mitochondria, especially important in post-mitotic cells such as neurons. Autophagy's protective role in neurodegeneration could therefore also be due to its role in mitochondrial clearance. Autophagy plays an important role in cellular maintenance in the myocardium. During aging, autophagic flux is slowed, leading to accumulation of damaged and toxic organelles and proteins, which can lead to cardiac dysfunction and heart failure. Experimental model confirms the importance of autophagy, which is induced in response to cardiac surgery in a number of animal models. Decreasing autophagy, for example, by decreasing the levels of autophagy protein 5, ATG5, leads to hypertrophy and heart failure in a mouse model. This suggests that whereas autophagy is required for myocardial homeostasis, one fundamental problem is that it is still difficult to monitor autophagy in a living organism, especially in a human. So future researchers are focusing on how to control the pace of it and monitor it accurately. There are literally many researches done which are directly indicating that autophagy has an impact on various diseases. I'll put some link down below. Remember we discussed about few diseases. The cause are mainly protein aggregates, oxidative stress, defective mitochondria, accumulation of damaged and toxic organelles and protein. So the question
is what would cause an accumulation of these misfolded proteins, which would be damage to the mitochondria coming from high levels of oxidative stress, usually from high insulin.
which lead us to the third part of the video, how fasting and autophagy are connected. Although there are many other factors which help in activating autophagy, but intermittent fasting is the easiest thing we can do. Fasting's anti-aging benefits come directly from the triggered autophagy when people stop eating for longer blocks of time. Fasting triggers two reactions. First, your body finds nutrition although you haven't consumed any calories, from where your body converts old junky proteins from within your cells into energy. This reaction takes place in lysosome of cell. Second, your cell experiences a burst in growth hormone production. Human growth hormone promotes muscle and bone health. This compound also pushes your body to empty its fat cells. According to Yoshinori, it frequently comes into play during starvation, allowing cells to survive periods of privation. Under nutrient starvation, AMP, activated protein kinase AMPK detects lack of nutrients, and the body is an adaptable machine. It is not gonna die immediately. It will send signal to the energy stores of the body. AMPK is going to activate the fuel sensor ULK1, which will then activate this pathway called autophagy. Then autophagy start its work, which means that new cells are going to eat your old and weak cells. It's going to promote the survival of healthy cells because you want to get rid of old and weak cells while making new and maintaining healthy cells. In his book, The Longevity Solution, Dr. James wrote that calorie restriction is the only non-pharmacological method of consistently extending lifespan and protecting against many age-related diseases. So, give your body a break, choose your calories wisely, and most important thing is choose your meal timings wisely.